thank you very much for the introduction. And thank you to all of you for the invitation to spend a few days at this wonderful conference at this very nice place. I'm currently living in Oxford. As the chairman has told you, and maybe some of you have seen in the news that we had a lot of rain. Actually, it was very flooded the last few months. I'm very happy to grasp some sun here in the south of Turkey. Okay, so what I'm going to present today is this big meta-analysis on the antipsychotics for schizophrenia. And before I start with that, I should briefly show our disclosures. They can be summarized such that I've received honoraria for lecturing and consulting with most of the companies manufacturing second-generation antipsychotic drugs. Before I go into the topic, I want to give you some introduction into what the meta-analysis is actually doing. For those of you who have a deeper interest in meta-analysis, I will have a second lecture this evening where I will explain a little bit more in detail how the method works. It's not a magic method. I think at least the simple ones are quite straightforward to learn. And this is also something that I usually recommend to younger psychiatrists, because if you do a meta-analysis, you really have to read all the studies very much in detail, and you automatically become an expert in the field. So I think it's a very useful method. Two issues are important. For a long time in medicine, we have focused very much on the p-value. The magic p lower than 0.05. This means something, this means statistical significance, and that's important. And the p-value is important, but we have all learned at some stage during our training that the p-value is only a measure of the probability of a finding. So it's a measure of the likelihood whether drug A is better than drug B or whether drug A is better than placebo. It has nothing to do with the magnitude of the effect. For example, the p-value depends very much on the sample size. If you have a huge sample size, then we can have a perfect p-value, p lower than 0.0001, but still the magnitude of the difference can be tiny. And this is where evidence-based medicine and meta-analyses come in, because once the p-value is established, once we know that the finding is not just a chance finding, but a real finding, then we talk more about effect size, which are measures of the magnitude of the difference between two interventions. And again, I will speak more in detail about that, how we calculate it in the evening. But the principle is very simple. So we just subtract one mean from the other, and then we divide by the good standard deviation. So for example, we have a schizophrenia trial, and at the end of the schizophrenia trial, with drug A, the PANSS total score is 90, and in the other group, the PANSS total score is 80, so the second group is better. Then we do 90 minus 80 is 10, and then we divide by the standard deviation. Let's assume that this is 20, so then we get an effect size of 0.5, meaning that drug B is half the standard deviation, half an effect size better than drug A. So by doing this, by subtracting and then dividing by the standard deviation, you get a measure in standard deviation units. And the next slide illustrates a little bit what that really means. This is the normal distribution curve of drug A, and the right one is the normal distribution curve of drug B. And you see that the effect size of 0.5, there are different terms for it. You will find buoyancy, accuracy. At the end, they're all mean the same. But effect size of 0.5 means that drug B shifts the distribution of response half a standard deviation to the right. That's basically the meaning, and again, I will talk more about it later tonight. But this is a very simple take-home rule that we should all know because it's really general medical knowledge. This is Cohen's rule, how we should interpret effect sizes. This is uniformly used in medicine. It's really just a rule of thumb, but Cohen said that on the average, the effect size of 0.2 is a small difference between interventions. 0.5 is a medium difference, and any effect size larger than 0.8 is a large difference between the interventions. And maybe try to remember these numbers a little bit because I will show you many effect sizes during my presentation. Now, how do we do that then in meta-analysis? In meta-analysis, basically, when we have, let's say, 10 studies on the same question, we take the 10 studies and then we want to get the average effect to know overall how good is the intervention. That's the concept. And here I have an example of olanzapine versus quetiapine for the overall efficacy, so PANSS total score. And these are the studies that we found, all randomized trials compared to olanzapine with quetiapine. We calculate the effect size for each of the single studies. And here actually you see the numbers, you see the means as I explained to you on the previous slide. And then we get the average of the individual effect sizes and get the average effect size down here. What you also need to know is that we also construct 95% confidence intervals around the effect sizes. These are the numbers in parentheses. And the same is displayed on this plot, which is called the forest plot. So here you see the individual effect sizes. Down here you see the overall effect size plus the 95% confidence intervals lined around. And the very simple take-home rule that you need to know to be able to integrate all these slides, they are called forest plots, the special term forest plots, is that whenever the 95% confidence interval is completely at one side, then we have statistical significance. So this is, for example, the case here. This one does not overlap with the y-axis. This one does also not overlap with the y-axis, and also the overall, the average effect size. So these results are themselves statistically significant because 95% confidence interval completely at one side means that 95% of the expected means would be in favor, in this case, of olanzapine. Olanzapine was the better drug in this meta-analysis. This one is not significant. Here you see it does overlap, or these ones, they also do not overlap. But another thing why this plot is very intuitive is that already graphically we see immediately what is going on here. So here, for example, you see a very consistent trend in favor of olanzapine because with this exception here, all the other effect sizes at least show the trend in favor of olanzapine because they were on the left-hand side of the y-axis. Okay, so we have statistical significance, but I told you, evidence-based medicine, we want to learn about the magnitude of the difference. So therefore, we also need to look at the average effect size, 0.23. And now we know the Cohen's rule, so you know it's a small difference between olanzapine and quetiapine. But I think, and I will discuss that later on, it can be important in this disorder like schizophrenia, that we have a lot of treatment resistance, many non-responders, and so on and so on. We have had a number of meta-analyses on this hot debate, second generation antipsychotics versus first generation, or atypical versus atypical debate, as they call it. And I just want to summarize a few of those, because then I want to introduce you into new meta-analytic methods, and then most of my talk will be on this new meta-analytic methods. So for example, my group, we had this paper in the Lancet with 150 double-blind studies and 20,000 participants. These are the outcomes that we analyzed, and we got a certain pattern. This is very busy, but this is not about details. I just want to explain the concept to you. So these are efficacy effect sizes. This is not only the average effect sizes. There was not enough space to put the single effect size of the single studies. But so these are the second generation antipsychotics in alphabetical order. And then these are the effect sizes plus their 95% confidence intervals for 4 domains of efficacy, overall symptoms, positive symptoms, negative symptoms, and depression. And if you remember the take-home rule, this can be summarized as basically 4 drugs: amisulprides, clozapine, olanzapine, and risperidone were significantly better than first generation antipsychotics, because now let's look at the confidence interval. You see here does not overlap. Aripiprazole, however, it does overlap with the x-axis, it's just turned down, but it's the same principle. Clozapine, of course, better with the highest effect size. Olanzapine, not different for quetiapine. Risperidone better than the other ones, but also all as effective as first generation antipsychotics. And we got the same pattern for positive symptoms and also for negative symptoms. because all the second generation antipsychotics are being marketed as being especially effective for the negative symptoms of schizophrenia, but for some of them, this has never been shown in a single randomized controlled trial. We also have head-to-head comparisons of second generation antipsychotics and meta-analyses on these, but now comes the problem. This is a summary of one of these meta-analyses. So whenever you see a number in one of these boxes, then there was at least one randomized trial comparing two agents head-to-head. And wherever the flash goes up, there was a statistically significant difference. When the flash is horizontal, then there's no significant difference, and the numbers are just the numbers of patients included in the analysis. So for example, this one here, aripiprazole versus olanzapine, means that olanzapine was statistically significantly better than aripiprazole. And again, if you compare it with the other meta-analysis compared to third generation antipsychotics, to a certain extent, a similar pattern emerges. So you see olanzapine shows up several times. Risperidone, the big surprise was that clozapine was not better than all the other second generation antipsychotics in this meta-analysis. We have discussed that in extension in the discussion section. We think that usually clozapine was underdosed in the trials, so there are problems there. But the main reason why I show this slide is that it shows we have one major problem in evidence-based medicine when there are many agents available. This is that we do not have at least one randomized trial for many of the comparisons. So for example, aripiprazole has never been compared with amisulpride, aripiprazole has never been compared with clozapine, thiamine, sertolone, and so on and so forth. So we have many gaps in the evidence, in the matrix of evidence. And then this is a problem for guidelines, because in this situation we cannot come up with clear hierarchies, which is the best drug, the second best, the third best, and so on and so forth. But that's ideally what we need for clinics to make our mind how we should choose among the available antipsychotics. And this is now where the new method comes in, the so-called network meta-analyses, also called multiple treatments meta-analysis. And the principle is very simple again. If we have trials that compare drug A with drug B and drug A with drug C, but we don't have direct comparisons with B versus C, then from A versus B and B versus C, we can indirectly estimate what B versus C is. And then we can use this indirect evidence, fill the matrix, and then produce hierarchies, which is the best drug and the second best, and so on and so forth. And that's what we did in the first network meta-analysis. On schizophrenia, you see that the number of trials is continuously increasing. Now we're already talking about 212 randomized controlled trials, all blinded. The reason is also because new agents, of course, come up, so we included 4 newer antipsychotics that have not been included before. And we looked at all the second generation antipsychotics plus the prototypal first generation compounds, which probably are haloperidol for the high potency drugs, and chlorpromazine is a typical low potency first generation antipsychotic. This plot is called the network plot, and I think it quite elegantly demonstrates what's going on here. The larger the circle size, the more participants were available for treatments. So you see most patients were in placebo groups, then haloperidol, olanzapine, and risperidone, sometimes treatments. Why do we still have placebo-controlled trials in schizophrenia? Is that unethical to withhold treatments, as we know that effective treatment is available? Well, the simple reason is that the registration agencies, the FDA in the United States and even in Europe, still recommend placebo-controlled trials, and therefore the pharmaceutical companies must do them. Now the first results, we go through the outcomes that we looked at in the meta-analysis, which was overall efficacy, but then also the major side effects of the drugs, and I will try to make some clinical comments and also some methodological comments about the various findings. So the first finding is overall efficacy of antipsychotics. And the benchmark, basically the y-axis, this is always placebo. So it's all benchmarked against placebo. And the best drugs in a given outcome, in all the slides, will always be on the top, and then it goes gradually down to the worst drugs at the bottom. Now you know the basic rule: whenever 95% confidence interval does not overlap with the y-axis, then the result is statistically significant. So you see that fortunately all the drugs that we have examined were statistically significantly better than placebo in terms of overall symptoms, being PANSS total score or Vickers total score. The hierarchy was also quite similar to what we had found in the simple pairwise meta-analysis before. So again, clozapine clearly on top, and then a similar group, emacipride, olanzapine, risperidone, haliperidone. Actually, this was also a very interesting finding that in almost all the outcomes, risperidone and haliperidone were very closely next to each other. And in some ways, for me, this gave face validity to the meta-analytic method, because as you know, haliperidone is just a metabolite of risperidone. The receptor binding profile is actually quite similar, so it made sense that in many of the outcomes they were closely together. And then it goes down, haliperidone, thiopin, somewhere in the middle. Something that is criticized, that has been criticized, and that we need to have a look at, is that the newest antipsychotics are phenantine, lurazidone, and iloperidone, which for schizophrenia at least are currently only available in the United States, at the lowest effect sizes. And there's a lot of debate about that, because people rightly argued, aren't these just cohort effects? These trials of these compounds have been produced very recently. So the argument is maybe the trial populations are nowadays different from the trial populations on the early second generation, clozapine, olanzapine, emacipride, and so on. And one very important component, so there's many compounds actually that we ruled out statistically, but one very important component is that people have seen an increasing placebo response in randomized schizophrenia trials. And this analysis by Kemp and colleagues illustrates this very nicely. So what you see here, this is just the PANSS total score reduction in the placebo groups, not in the drug groups. And you see that the earlier studies, so the risperidone studies published around 1993, there was no placebo response, was on the average 0, while the latest antipsychotics, haliperidone, azathioprine, difepanox, there was a big placebo response, about 10 points reduction just in the placebo group, and therefore the argument is if there's a lot of placebo response, it is difficult for the antipsychotic to be much better than placebo. And I think there is some truth about that. We ruled it out statistically, so one thing we did is that we added, we reran the analysis without the placebo-controlled trials. We also added publication here as a moderator, as another component in the analysis, but the analysis was robust to these findings. So actually, I think there is always an argument how much can we really do with statistics to find such problems. But I also think that there's also feedback that I get from many audiences like you, that overall what we saw in the meta-analysis is also what people see in their practice, that there are differences in the efficacy between the antipsychotics. One thing that is also interesting, where does this increase of the placebo response come from? I think the most compelling argument is that nowadays very often there are clinics who professionally do clinical trials, and they recruit their patients by newspaper advertisements, for example. So they attract patients who have been called healthy volunteers or professional patients. So these are people who have some benefit, some financial benefit also to include in the trial. For example, they get reimbursed travel costs, which for a welfare recipient can be quite much already, or they get the trial medication for free. But these are not the ideal highly acute patients, which will probably not show a placebo response and which will show a big drug placebo difference. There's also many other compounders that we try to rule out. only when the haloperidol dose was higher than 12 milligrams a day, only then was the difference compared to second generation significant, not when they used lower than 12 milligrams per day. The argument was if you high dose haloperidol to patients, they all have extrapyramidal side effects, and these extrapyramidal side effects then look like negative symptoms, and then we get an artificial superiority of the second generation antipsychotics that do not produce these extrapyramidal side effects. Well, we ruled it out again, as I said, statistically, and we controlled for the haloperidol dose, but did not get much in that regard. Another big problem in schizophrenia trials nowadays is that it seems that the dropout rates are continuously increasing since the early trials in the 1950s and 1960s. Nowadays, very often the dropout rates in randomized schizophrenia trials are higher already in the acute phase, higher than 50%, and this poses an enormous problem to the statistical analysis of such trials. Next outcome, all-cause discontinuation, dropout for any reason. That's an outcome that is used very much, especially in so-called effectiveness studies, because it's quite elegant. The main reasons why patients drop out of clinical trials in schizophrenia are either the inefficacy of the treatment or side effects. So you have a composite of efficacy and the tolerability of the treatment, which is something that we want, which is interesting, and which therefore some people have also called acceptability of treatment, because again, treatment that is not efficacious is not acceptable for patients, and definitely, of course, treatment that has many side effects is also not efficacious, is also not acceptable for patients. So I think it makes some sense. The problem is that people very often think that acceptability is a side effect, is tolerability, but that's exactly a problem in the schizophrenia literature, because you see we get the same hierarchy more or less as before. So the same five again on top, amiloxicillin, the lansing, clozapine, haloperidol, risperidone. So that's surprising. But the reason is that the main reason in schizophrenia why patients drop out is inefficacy of the treatment and not the side effects. This is, for example, different in antidepressants, so it's the other way around. But that probably explains why we get a similar pattern in terms of all-cause discontinuation and in terms of overall efficacy. Not exactly the same pattern. So, for example, you see haloperidol was the worst drug in all-cause discontinuation, while it was in the middle for efficacy, and the reason for that is probably the high side effects of haloperidol. So there are other terms that are discussed. Dropout for any reason. The problem here is there's no clinical meaning, that's why I don't like that. All-cause discontinuation, I think it's pretty good because it's a neutral term. I think the best way how we should call it in the future is retention or staying in treatment, because this really shows the importance of the outcome. It's not so much that patients drop out. What we want is that patients stay on the treatment, that they are complying, that they are adhering with the treatment. So this is my preferred term for that reason. Just some quick examples from other big trials, just showing that what we call an all-cause discontinuation was also found in big trials that we have excluded for various reasons. So, for example, the CAPE study, this industry-independent study from the United States, which cost $50 billion, was included because it was a long-term trial. We just look at phase 1, so 1,500 patients were randomized to olanzapine, the type, risperidone, ziprasidone, and fantasy. And then there was phase 2 and phase 3, but it's just a quick look at phase 1. So the best part was olanzapine. The surprise was that fantasy was not worse than risperidone, but then the type and ziprasidone were worse ones, although most of the results were not statistically significant. But I think we can use it as an argument that the pattern overall, again, was the same. In phase 2, then patients who had discontinued phase 1 could be re-randomized to either clozapine in one arm, all the other second generation, and again, clozapine was the best one here. Although there was one bias, this is that these three were given double-blind, while clozapine for ethical reasons was given open label, so still there's some problems there, but the pattern was pretty similar. There's also a European first episode study, effectiveness study, long-term study, which we could not include because it was open label, but it's very important trial, the UFES trial. Here they compare low-dose haloperidol with antipsychotic olanzapine, and ciprostol, and all that. First episode patients, again, more or less similar pattern emerge. On the left-hand side, you see the survival curves, on the right-hand side, you see the actual numbers. So olanzapine and antipsychotic, again, were associated with the lowest discontinuation rates, and then these are the other drugs. So again, a certain similarity with what we had seen in the meta-analysis, although the studies were excluded. Next outcome, weight gain. We all know that this is a very, very important outcome because the long-term consequences can be dramatic, diabetes, and maybe even longer term strokes, heart attacks, and so on and so forth. And the pattern that we got was not unexpected. That was basically something that we knew already before. Haldol, ziprasidol, Duracell, this American drug, aripiprazole, antipsychotic, azathioprine, these were the good ones. Then the middle group that I think starts somewhere around haloperidol, and then the risk ones, clozapine, sozapine, and olanzapine, as we all know, so very important outcome, of course. I also like, however, to use this slide to show something about this typical versus atypical debate, because at some stage people said, well, all the second generation, all the atypicals are free of EPS, and nowadays we know that this is not the case. A few slides further on. But then other people said, but all the atypicals, all the second generation, and no first generation, no implication, produce a lot of weight gain. But both are not true. We see that some of the second generation, ziprasidol, aripiprazole, antipsychotic, are quite free of weight gain, but some old first generation antipsychotics are also weight gainers. Basically, all the low potency first generation antipsychotics, here we only looked at chlorpromazine, but I think we can also be said about all the other low potency first generation drugs that are sedating, are also weight gaining antipsychotics. So even also in terms of weight gain, this dichotomy to typical and atypical antipsychotics does not really hold, and this will be one of my major conclusions that this typical atypical classification does not make a lot of sense. I've presented you the data in terms of effect sizes. There's a famous, now it's old but still valid, meta-analysis by Allison and colleagues who did it in kilogram within 10 weeks, and you basically get a similar pattern. So olanzapine goes to be 4 kilograms approximately within 10 weeks. I think that's also important because I think all of us also know patients who have gained much more weight during longer duration. Extrapyramidal side effects. We use anti-Parkinson medication as a proxy because this is a measure that is objective and that is very consistently reported in the trials, while the single extrapyramidal side effects, there are variations in definitions, and it also depends on the doctor, whether the doctor really wants to put down the patient who has dystonia or had Parkinsonism. So it's more subjective, while the anti-Parkinson medications are very objective. Measure, and again, I think the results that we got made a lot of sense. Clozapine the best, then a group of Sertidol, olanzapine, tyripine, aripiprazole, maybe a little bit too high here, but these anesthesia phenomena. We know that this is the main problem of aripiprazole. Then a bigger group, and haloperidol, clearly the worst ones in terms of extrapyramidal side effects, even when we only included haloperidol doses lower than 7.5 milligrams, they still haloperidol was associated with really more extrapyramidal side effects than all the second generation antipsychotics. A few clinical remarks. Some of you may have spotted that clozapine actually even reduced EPS compared to placebo, I think there are two major explanations. One is a methodological one. Very often patients are pre-treated with antipsychotics before they enter the trials. Then there's a very brief washout phase, which due to ethical reasons can be shortened to 2 days. And that's not sufficient to wash out all the previous EPS from haloperidol and all the other drugs that patients have had. And then we get overlap, carryover effects from the previous treatment. And then a drug that is basically free of EPS, like clozapine, might suppress these carryover effects from the previous treatment phases. One explanation. The other explanation is that it's pretty well established that about 10 to 20% of patients with schizophrenia who have never been treated with antipsychotic do have movement disorders, do have extrapyramidal symptoms. And again, it could be that a very good drug in terms of EPS like clozapine suppresses these extrapyramidal symptoms. The other thing that I also already mentioned is that not all second-generation antipsychotics are free of EPS. I think we know that risperidone, haloperidol are the classic examples. And chlorpromazine, the low potency one, was a little bit low here, but still, all the low potency in this also always produce severely fewer extrapyramidal side effects than the high potency ones, such as haloperidol. So, also in terms of EPS, this classification in a dichotomous way, which we typically make with antipsychotics, is not really valid. Prolactin increase is an outcome that can be very important. It is associated with osteoporosis, with sexual side effects, and there's also debate whether it might promote breast cancers. Although the latter theory is really a theory, this has not been proven to as much as I know. Peter Haddett has had a nice video on this question, and he's also lecturing at this conference. So I hope that you agree with what I'm saying, so there's a debate about that. But this is not a proven association. Again, the hierarchy is not unexpected. Aripiprazole, the best one. Remember that aripiprazole is a partial dopamine agonist, which in situations where there is not enough dopamine pass through, it actually activates the dopamine receptors, and therefore might induce phenomena like prolactin increase. You see that it almost statistically significantly reduce prolactin compared to placebo, and then it goes down. And really very interestingly, one thing that risperidone and haloperidol on the one hand, they basically have the same effect size. I think that the pharmaceutical company at some stage did make it very clear that haloperidol is really the same in terms of prolactin increase than risperidone. And the other important thing is that it's really a very big effect size. Effect size of 1.2, 1.3. This is very rare in psychiatry. I rarely see such high effect sizes in psychiatry. They are even worse than haloperidol, which was only the third worst in this regard. I need to note, however, that we didn't have any data on antipsychotics, which is also a drug that is very much known for leading to a lot of prolactin increase at the same level as risperidone and antipsychotics. Some more clinical notes about that. Prolactin by itself is just a laboratory value. We should not forget that. So usually I would not recommend, and I think that also most of the guidelines do not recommend changing the treatment only because the prolactin level is increased. We should only do that when there's really a side effect, and then we find, maybe we confirm that prolactin is the reason for the side effect. But we should not just due to the laboratory level change the treatment. So this is one important thing to consider. And the other one is that, in my opinion, the link of prolactin increase and side effects such as galactorrhea and amenorrhea in women, this is very clear. This is a very clear link. A more complex, but maybe more important phenomenon is the lack of libido, which also has to do with prolactin increase, but it's not the only factor. So, for example, also the negative symptoms of schizophrenia play a very important role in terms of lack of libido, so this is more difficult to understand. QTC prolongation, this is a side effect that attracted our attention when Sertanol was introduced in Europe and actually never made it to the American market due to the QTC problem. And therefore we were actually quite surprised, or not surprised, but it was interesting to see that the effect size is really actually quite high in terms of Sertanol and QTC prolongation. You see that the effect size was 0.9. Another thing which was also used as emasulfite, which as you know is a selective dopamine receptor antagonist. So many of us, including myself, have used it for a very long time, especially in patients who had cardiac problems, because I thought this is a selective dopamine receptor antagonist, so it shouldn't have a problem in terms of QTC prolongation. But our meta-analysis is not the only paper. There's also two other recent papers with other methods. These were naturalistic and also showed that QTC prolongation with emasulfite is actually quite a frequent phenomenon. So nowadays I do not use emasulfite anymore in patients with cardiac problems. I think this situation is more it's a drug that should rather be avoided. The third one is ziprasidone, and then you also have good ones, of course, lurasidone, aripiprazole, paliperidone. Haloperidol is the first one that produced statistically significantly more QTC prolongation than placebo. You see that the effect size just does not overlap with the y-axis, but it's also a trivial effect size, 0.11, so that's certainly negligible. And then there are many drugs, some with a little bit small to medium effect size, what that mean. But I have heard several lectures from cardiologists about this problem, and what I took from these lectures is that in psychiatry we are a little bit over-concerned about the QTC problem. So we should be careful, of course, but it seems that there are many drugs that are used in medicine that have the same phenomenon, and doctors do not care so much about that. What do the clinical data really say? Ziprasidone, as I said, was the third worst drug in this regard. So Pfizer had to do a large randomized trial, 18,000 patients randomized. It's a massive number, a major achievement in this trial. Just to look at the question whether ziprasidone produces more cardiac mortality than comparator olanzapine. This is the so-called Zodiac study. Unfortunately, there was no difference in terms of cardiac mortality between ziprasidone and olanzapine. So therefore, ziprasidone can be prescribed without specific ECG measurements, for example. Unfortunately, this is the case for Sertanol, where they also had to do a large randomized but then naturalistic study, the SCOP study, with almost 10,000 patients. And there was no difference between risperidone in this case and Sertanol in terms of all-cause discontinuation, but the cardiac mortality was higher with the Sertanol group compared to the risperidone group. And therefore, if you do Sertanol nowadays, you need to do all these ECG measures and so on and so forth. There was also an interesting signal that maybe suicide mortality is reduced with Sertanol, but that was not statistically significant. Unfortunately, the company is not followed up because the compound are not interested in that. And then finally, sedation, which I think is a very important outcome for patients because patients very often complain about sedation. In research, it's neglected. Usually, it's only measured by open interviews, so basically, we don't have the rating scales or objective measures for that. It's basically the question that the patient in the trial really complains about sedation in an open interview. Therefore, we don't have good data about it, but still, I think that the hierarchy that we got was actually quite plausible. Emasulfite, the selective dopamine antagonist on top. Paliperidone was a little bit better than risperidone here. I think the reason might be that paliperidone has this extended release mechanism, so they're not the same plasma level peaks after ingestion as we get them with risperidone. It's a more stable plasma level, and this might explain why it's less sedating. Sertanol has always been advertised as being effective antipsychotic without a lot of sedation. Aripiprazole, I think that's also not a very sedating compound, and then it goes down. Olanzapine, thiopin, more sedating. Ziprasidone, that was a bit of a surprise. I wouldn't have guessed it so far down. but then clearly the worst ones, all the low potency typicals, chlorpromazine, zolpidem, clozapine, of course, very sedating compounds. Most of you will know zolpidem is the Japanese drug, and Japanese colleagues told me that they especially like to give it in acute mania due to its very sedating profile. So again, I think they're very reasonable hierarchy. So what should we do with all these data? And I think the good news nowadays is that compared to now 20 years ago when I started in psychiatry, we're in Germany, basically we used two drugs. We started everybody on haloperidol, and then if a patient had EPS, then we switched the patient to parazine, which is a chlorpromazine like low potency drug. That's basically all we did. I think nowadays we have much more variety with many different compounds, with partly different mechanisms of action, with very different profiles, I think, in terms of mainly side effects, but also to some extent in terms of efficacy. And I think that we now can use this data to adapt the drug choice to the needs of the individual patient. And how should we do that? I think one good way, and this is a new concept, so I think it might be interesting for some of you, is this concept of shared decision making, which can be best explained by explaining its extreme counterparts. What we are currently doing in medicine in general, I think, is still a pattern listing model. So basically, the doctor knows everything, he's the expert, he chooses the medication for the patient, and then the role of the patient is to be a good patient and basically to be compliant with the medication and to follow what the doctor says, as it's explained here on the cartoon. The opposite, what is used in some countries, especially when we go to tropical countries and we want to inform ourselves whether we should use a vaccine for malaria or whatsoever, then we have this concept of informed choice. So basically all the information is available, for example, on the internet, or there are special agencies that we can call, that we can consult, and they say these are the data, but then it's really up to the patient to make a decision whether he or she wants to take the vaccine or not. So the responsibility is fully with the patient. And shared decision making is somewhere in between. Ideally, it should work like that before the decision is made, the patient is informed about the different options. So this is, for example, a slide from a trial that we have conducted in Germany. We produced a little document in which we inform patients in very simple terms how medication can be taken, either as tablets or depot medication, for example. With such a thing, the patient is referred, and then the patient goes to a decision talk together with the doctor, and then they come up with a joint decision. So shared decision making does not mean that all the decisions with the patient, it's still the doctor and the patient arguing about what the best way is. And the hope, of course, is I think there are two main factors. One is at the end it's the patient who must take the medication, so we should involve patients. And the other hope is also that by involving the patient, maybe the compliance and the adherence to the medication gets also better. These are the most important concepts. And actually, you don't need to make formal shared decision making. I think it's an interesting concept. You can, of course, not do it with all the patients. Somebody is highly agitated, violent, doesn't want to take any medication. Of course, shared decision making does not work. But for me, it's mainly an attitude. So how much this has changed my practice is that usually I don't do formal decision making with booklets and so on and so forth. But very often there's no good answer. So the answer is, well, either you take that drug and you have weight gain, or you take that drug and you have extrapyramidal side effects, or you have a third drug and you have sexual side effects, or you take a drug that is very good in terms of side effects, but the efficacy is not very good. And if possible, then sometimes I would like to involve the patient and ask patients, well, in your situation, what is your value? What is most important for you? And then you get a better decision sometimes. So to sum it up, I think that there are differences between the antipsychotics, mainly in side effects, but also in efficacy. But the side effects differences are clearly larger than the efficacy differences. I think that the hierarchies that we found in these domains allow clinicians to adapt drug choice to the needs of individual review also made very clear that this dichotomy in typical atypical antipsychotics, first, second generation, does not make a lot of sense. I think they are all antipsychotics, but with many of them, and we should use them to give the best treatment to our patients. Thank you very much for your